Hi everyone, welcome to another session of Sybeco. I'm Farzaneh Najafi and we're hosting Yuan Zhang today. Yuan completed her bachelor's in biology at Nankai University with Dr. Zhong Tao. She studied in her bachelor's the treatment of depression using repurposed drugs in rats. Then she started her PhD at the Ion Institute of Neuroscience with Dr. Ning-long in Shanghai, studying decision-making behavior in cortical circuits. And now she's a postdoc in Dr. Zhu's lab studying dendritic integration during flexible behavior. She has a sustained interest in the mechanisms of consciousness, including attention, perception, and brain-machine interfaces. Welcome, Yuan. Please go ahead.
Okay, thank you. Thank you for your introduction, and thanks just for giving me this opportunity to share my work with all of you today. So I will talk about the dendritic computation for Ruby's flexible categorization. Mm. So what is Ruby's flexible categorization? Uh, uh, the behavior flexibility is very common and important in our daily life. So one of the most famous paradigm is the Wisconsin card sorting test. So in this task, the subjects are supposed to classify the cards by different rules. For example, like color, shape, and number. For example, this card should be classified into this fourth group by color, but it should be classified to the first group by shape. So when the rule changes, the subjects need to use the feedback about their correctness to adjust the rule and then to adjust their choice in the following trials. Um, so to simplify this question is for the same sensory and a different rule, they need to make different choice. Well, how single neuron do this computation is still not clear, so that's we want to address. And there are 5 pyramidal neuron would be very good candidate to study this question within a neuron, since they are key computational units. The layer 5 pyramidal neuron is the largest neuron in the neural cortex, which can integrate information from an entire cortical column, and they also can compute the information from the feedback information from other brain regions, and then they can make the output to distant targets. And previously, the electrophysiology studies show the distal dendrites here where the calcium spike majorly initiate. And for the basals, where somatic sodium spike initiate. And they coupled by the apical trunk. And functionally, the tuft can integrate prominent long-range top-down information. And for the basal part, it's mainly integrate bottom-up, local, and other inputs. And this could be influenced heavily by calcium spike in the tuft. And trunk modulate the coupling between the two. But what the behavioral variable are those top-down and bottom-up information represented for? And especially how the top-down information contribute to behavior output at subcellular level is still not very clear. So to clarify our question, we want to examine the dendritic integration in a flexible categorization task where we need the top-down information, the rule information, combined with the bottom sensory information, and then make the correct choice. So our first question is, we want to know whether there is compartmentalization in the different dendritic compartments, whether they represent for different task variables like sensory rule and choice in a behavior involved verifying neurons. And the second is if there is compartmentalization and how dendrites integrate those information to contribute to their neuronal output. So to answer this question, we trained the head-fixed mice to learn an autonomy-based flexible categorization task. And each trial starts with a pure tone stimulation. If a low frequency tone played, mice need to lick left to get the water reward. And if it's a high frequency tone, the animal need to lick right. So the rule changed, and the rule one for the three middle tones, the animal need to lick left to make a left choice to get water reward. And if it's under rule two, for the same sensory stimulus, the animal need to lick right to get the water reward. So the three middle tone are the reversing part.
So
And
just
for
to make
the
sure, the rule is the category boundary. That's what changes.
Yes.
Right.
Yeah, correct. Yeah. And for the end trials, they are non-reversing trials. That is under different rule. They don't change their choice. And for an example session, we got reversing trial and non-reversing trials interleaved given to the animal, and the rule was block by block given to it.
How
So
long
that's
was
our
each
task
block?
design. Sorry?
How long was each block?
How long? It's about more than 150 blocks at least, I think. We have a threshold. Actually, the animal change the trial length for each block is flexible. It's based on the animal's performance.
And about what? Like it's about tens, 50s?
Like 150 to more than 200, and yeah,
But
like
then you
this.
said you have 150 blocks in a session?
Oh, no. Trials.
Trials. Okay. And then how many trials do they generally perform?
Generally, for the fastest animal, maybe just like 20 to 30 trials, including non-reversing. Yeah, they can do it. They can do the reversing.
I
Yeah.
guess I'm confused. I'm sorry. So you're saying each block, I understood it's flexible, but you said how many trials are on average in each block?
Oh, each block is like more than 100 to more than 200 trials
Okay,
for
thank you.
each block. Yes. So
Thank you.
totally the animal usually do like 600 trials.
Okay,
Yeah.
thank you.
So it will get 3 to 4 blocks each day, each session.
Awesome.
And so this is our task design. And when mice learn that behavior, we can get these two psychometric curves, which is rightward choice performance as a function of the frequency with middle choice separate as is shown here. So with this behavior, we can having the non-reversing and trials. They share the same sensory choice, but rule is different. And also we have the middle reversing trials. They share the same sensory, but the different choice based on different rule information. And we can get the animal's subjective boundary here. And when mice learn it, it can separate this two rules based on their own boundary. Means the animal learned this task. And when animal learn this task, we combine two photon imaging with this behavior to study the dendritic activity. We implant a 2 to 3 diameter chronic window in auditory cortex. So this is an example mice doing task during imaging. And here's an example stack of imaging field with GCAP6 expressed. And we can see the apical dendrites. So here, these are the apical dendrites
And
here.
this is layer 1, where layer 2, 3, where are we here?
The apical
this is
dendrites.
this.
The apical dendrites is from the pia, from layer 1, and go down and to the somites stay in the layer 5.
Nice.
So apical dendrites in layer 1, the tuft reach to layer 1, and then goes down, and we can see the single point. That's the trunk. The trunk is like more than 100 to about 400 depth, and then the soma is around 500 to 600.
Is it virus expression or transgenic animal?
Oh, it's virus. I injected a retrograde virus.
Okay. Retrograde where?
In the superior colliculus, because in my previous work, we found the auditory cortex to SC projecting, this projecting their 5 neurons is engaged in our categorization task.
I see. So we are looking at layer 5 neurons that project to the superior colliculus all along their dendritic tree, along the cortical layers.
Yes. And this layer 5 neurons in auditory cortex.
Yeah. Okay, thanks.
So this is an example.
I got a question here.
Okay.
If you can go back to previous slide. So it looks like you guys are doing a Z-stack scan in that one.
Oh, that is just a Z-stack after behavior. We are not doing it during behavior.
Okay, that's what I was curious. Is like, okay, are you scanning through the Z dimension throughout the entire session, or is this something like? Because I was thinking like that's something that I would like to do is like to get a Z-stack scan so we can actually map ROIs to the true morphology and say, okay, which neuron has which contributions. I didn't know if you were doing that throughout the session.
Oh, so far we are not doing it during the whole session. It's just after the mice doing the behavior task, and we do a Z-stack to know where our imaging field is. Yes.
I see. That was a good question then. So I didn't also I thought throughout the task you were doing volumetric imaging. Okay, and then for the actual behavior, you were focused on which plane?
Yeah. So for behavior, we have fixed plane to image because we have different trials, and our speed cannot allow us to do a whole neuron imaging so far. So this is an example field, example tuft field. This is the estimate depth in the dendrites. So for each session, we fixed our imaging in one plane, in one depth. So this is an example ROI in tuft field. So here each line was a trial, and the color here means the intensity of the calcium activity. And the correct trial of a session was separated by different frequencies and different rules. And the trees were averaged by each frequency and each rule. And to make well control of other mixed information, we test the rule-related activity in the non-reversing trials only. So because these trials, they share the same sensory and the same choice. And also, those are all correct trials. So they share the same reward. So here you can still see this difference. Only difference is rule is different. So you can see a significant activity separation in the non-reversing trials. So this is we quantify this difference using a ROC analysis, and we found this is significantly rule information.
How long is the stimulus?
Sorry?
How long is the stimulus?
How long is the stimulus? Oh, you mean the sensory stimulus, the pure tone? 300 milliseconds.
And then it's not a working memory task. So you present it for 300 milliseconds, and then after that they make their choice.
Yes, it's not a working memory task.
And the spouts, they can lick during the stimulus. Are the spouts available?
So you can see the first white line. This is the stimulus onset.
Correct. And then I just want to know if they can lick during the stimulus, or do you make the spouts unavailable during the stimulus?
Oh, they can leak. Oh, we not prevent them from leak. Yeah, they can always leak. Yeah, so we don't have a delay here.
Okay. So then there could be leaking confound here. I suppose you have lots of supplemental figures to address that, but I'm saying you would want to make sure that between rule 1 and rule 2, you don't have difference in leaking.
Oh, yeah. Oh, that's the next point.
Sure.
Yeah, so for rule 1 and rule 2, they are all they are all same sensory stimulus, and they make the same choice, and they all correct trials. So for the same correct trials, they really not have much difference in leaking.
Sure.
Yeah. So but yeah, we can add that analysis here.
The other question I have, for dendrites, how do ROIs look like? Like the figure you're showing there with that red and the yellow. So is that one of the ROIs? One of the that you is that, like that counts as one? That segment counts as one dendrite?
Yeah, this is the example I showed here. And previously we used hand draw the ROI. And so now we have C2P. So we can use C2P to draw the ROIs based on the activity.
You say manually selected with C2P?
C2P not manually. It can automatically draw based on activity.
Yeah. Okay.
So even dendrites, just change the settings. Yeah, so they can automatically draw.
Thanks.
And so for this slide, we found there is rule information in the dendrites. And then we go deeper. I'm stuck here. Okay. And then we go deeper to the trunk. Trunk's position is like here. And so for this example, ROI, we still found there is separation in non-reversing trials. Means this is the rule information. And then we go to the soma. Soma is explained in layer 5. But when we reach to this example, soma, there is no difference in non-reversing trials of this example, soma. So for this soma, the rule activity was left here. So these are example neurons, and then we summarize all the ROIs we have, and quantified by the AUC analysis, we calculate the significant rule selective ROIs proportion, and we found dendrites have more proportion coding for rule than soma, and for the and this is for the proportion, and this is for how much the information coding is, and we found the AUC value for dendrites is also significantly.
Question. So do you do this across sessions? So it's not the same session that you have all the three compartments, right?
Yes, across sessions.
Okay.
Yeah, so I just put all the sessions together.
And then
Let's
behavioral performance, you have control for that? That's similar across the sessions?
Oh, behavior. Yeah, we did not control for the same behavior performance.
Right, because that can be a confound.
Oh.
But maybe it's not because you're recording from trunk that you don't see as much rule information. It's because they just didn't pay attention as much in those sessions and the performance was lower. So it's important to make sure that the behavioral performance is similar across these different sessions.
Yes, the correlation between behavior performance and the rule information. Yeah, that's a good point. Thank you. I'll add it. And so here we found there's more rule information in digress than soma. And then we want to know what is that dendritic rule information? Yes, what the animals think about. So there's some internal variables the animals thinking. So we fit a BSRL model. It's previous done by our lab member Liu Yanhe, and with the same task. So this model is it's to so for each trial when the sensory stimulus comes and the animal make choice and there is outcome to tell the animal is correct or right. So and then the animal can have a prediction error. And not just update by the trial and error, and she found the animal can also influence which rule, like high boundary or low boundary, the animal was in. So with this rule information, the animal can it can better fit the animal's behavior. So here we also quantify this rule information with our data. So this is for the this scored activity of the rule selective ROIs selected by the AUC, and the x-axis with the boundary estimation value from this model. And we found they are significantly correlated. So, which means we found this rule difference, and it could be explained by the animal's internal estimation of the boundary. And so far, we just stay in the rule information. And then we want to see other behavioral variables. So
could you go back? Sorry, what was the conclusion? So I understand that the line slopes are different. Like in the soma, the magnitude of the W doesn't affect the activity. So then that's your way of concluding that then the neuron is not, the soma is not encoding the rule. Otherwise, you would have expected to see some correlation. Is that right?
Yeah, the soma is has less rule information.
Right. Okay.
So also one quick question. the boundary estimation, I think, would change during the block as the animal is more familiar with the block. So if you compare this for the first half of a block and the second half of a block, does it change?
Oh, yeah, we have a, oh, I think here I don't have that slide. Yeah, so you mean for each behavior session, it has early trials and late trials, right?
In each block, yes.
In each block. Yeah, so far we don't have it, but we have this correlated value, like because for the boundary estimation value is trial by trial calculated. So each trial it would have a estimation for the boundary, and there is each trial there is a calcium activity value. So when the boundary because each trial is change the boundary estimation and the changes calcium activity. So here at least we can say the calcium activity and the boundary estimation is trial by trial correlated.
Okay, so your higher boundary estimation values would be towards the end of a block when the animal is more confident.
Maybe.
Okay. So the activity goes up.
Similar.
Sorry.
So go ahead.
Go ahead, Dieter. Did you want to?
Yeah, I was just wondering that the activity goes up and up then during the block. If the calcium activity goes up with boundary confidence and the boundary confidence goes up with the block, then the activity goes up during the block as time goes on.
Oh, you mean whether the activity always goes up?
During as the block progresses, yes.
Yes. I used to plot for the single sessions. Yes, for some blocks it looks like it, especially for the neuron has its own preferred block. So usually the calcium activity gets higher and higher in the neuron's preferred block.
Thanks.
Okay.
Right, so you'd see your weight variable shift there throughout the block.
Sorry?
I think it's like similar to saying you would see your weight there on your rule weight should shift throughout the block. And that's a similar question that that I have is did you look at how the activity shifts, not just throughout the block, but at block transition? Because a few slides back when you're showing the different activity whenever in each area when the mouse is operating on rule 1 or rule 2 and how those differ. I was curious, do you see that? Is this, a very, is a fast shift when the mouse realizes the new category boundary as they transition from one block to another? Or is it this gradual shift?
Oh, you mean the shift speed?
Well, like if you essentially, but like a few slides back where you're showing the rule 1, rule 2 raster maps. Like, do you see, because I mean, it's not instantly on the first trial of a block transition going to shift activity like that, right? There would be, some number of trials before you see, okay, mouse is operating on category boundary 1 until they adapt to category boundary 2. So there should be, like where you're showing the green and orange average DFS, like that should be, like it's not going to jump from one to the other. I didn't know if you looked at that. Like, okay, when we go from one block to the next, how many trials until we fit to what the activity pattern indicating each category boundary is.
I think for
Does that make
each
sense?
animal, oh, sorry.
Does that make sense? Do you know what I'm asking?
Yeah, I think so. I think that's a good question. Um,
So all
so
of the questions, Yuan, are both Didier and Tim's questions are about the trial by trial change in this correlation, basically, or in the activity. So whether it's about at the time of transition, do you see that, for example, some dendrites encode that change all of a sudden? Like, well, it's like a gradual thing that they realize about the category boundary shift. Yeah, so I think both of the questions are just like along the same line of trial by trial activity.
I think
Which
for
you said
the transition part, maybe there is some trials show very big prediction error signal. And
yeah,
then
but
the
you have those? Have you quantified those?
I don't have it so far. Yeah.
Okay, that's okay. I
I
had a
think
quick
that's good.
question and then we should move on. So how many ROIs do you get in one session?
How many ROIs? Oh, that
Yeah.
depends on the field.
But about
About for Soma drivers in different fields uh
because your
like
field of view looked to be smaller than what we would do. So it seemed to be only about 100 or something microns. So I don't think you have too many ROIs per session. So is it in the order of less than 100, perhaps?
less than 100, yeah, for each session.
Okay, that makes sense. All right, yeah, let's keep going.
But for Taft, it would be more because it's more dense, and for trunk and Soma, because I choose a relatively sparse area, so not too dense.
Okay.
Okay. So then we look at whether there is compartmentalization. So we use a GRM model. And here you see the sensory choice and the rule information. Here's the rule information using the boundary estimation. And we found the dendrites show less sensory information than soma, while dendrites show more boundary estimation information than soma. Well, choice, because we think choice is the result of a integration, so so here is not much difference in choice. So it's like a sensory with the rule come here, and then they integrate into a trace output. So that's our neuronal hypothesis. So so far, we found the difference in compartmentalization, behavior variable coding, but this is for the dense labeling neurons. We didn't follow the same neuron. So then we want to see whether
Question, sorry, and this is aligned with what Tim was asking. So in the previous slide, your model does have a prediction error component. Did you quantify the relative contribution of that as well?
oh yeah, it's very small. Yeah, we have that figure in the paper, but I didn't show you here. It's very small.
It's
So
very
it's
small and
not
it doesn't
much.
change across compartments.
Well, because here we use all trials. So for the transition trials, it's a very small proportion. So I think that may be one of the reason we see very small prediction error signals here. So I think a better way to quantify is to pick up the transition trials and then look at the prediction error directly in that few trials.
That's good. Thanks.
Then we want to see whether this phenomenon is consistent within neuron. So here we use piezo when mice performing the task. So piezo is just about 7 hertz. is much slower than one plane imaging. And so this is the example dendrites, and this is the example soma. And so for this neuron, this is the trunk of this neuron, and this is the soma of this neuron. And so for the trunk, so this is the activity from the trunk, just like the activity I showed before. So we can see there is still separation in non-reversing trials. This ROI is have the rule information here. Well, for its own soma, there's no separation. It's not significant anymore. So within neuron, we can still find dendrites have more rule-related information than its soma. And then we
Okay,
summarize.
sorry, could you go back? I just realized I had to know. So for soma, I'm seeing that during the reversing, like for those reversing tones, there is a separation.
Oh, reversing tones, yes. Because reversing tone, their rule and twice are different. Their twice are different.
What is different?
Twice. Their left or right twice is different.
Right. Right, because the choice is different. So you're saying that
The big separation in the
yeah,
reversing tones.
your voice is a bit cut enough. Okay, and then one other question. How do you make sure that those trunks belong to the same soma? Is it by some correlation?
We have a Z stack, and we can trace the same neuron.
So it's just visual, like observation?
Oh, yes. And we can also have correlation, like they share the same, they share the very high correlation of their activity. Am I stuck here?
You're good. I guess we can just
Oh,
should just move to the
okay.
next one. Yeah, we have you. Did it move? Have you moved your slide? Because it's stuck
Oh,
for us.
okay.
No,
Oh,
yeah, it's
it works.
yeah.
Okay. So this is a example neuron, and then we quantify all the single neuron we can trace. So we found still the sensory information is higher in soma than dendrites, while rule information is more in dendrites than in soma. Trace information is not much difference, and now we can be sure that we found the compartmentalization of different decision variables. But then whether the feedback rule information can contribute to the trace output at neuronal level. So
A quick
here,
question. Did you examine trunk and tufts separately, or when you say dendrite, you just mean either of those?
Because previously I showed the trunk and the tuft information, they are very similar. So so we just think the apical part activities are similar, and they are different to their soma. Then we want to see whether there is causal effect for the dendritic Ruiming information for the trace output in the soma. So we know the dendritic targeting SST enriched cells in the cortex, they could downregulate the dendritic activity. So here we try to label those deep layer SST neurons and perform imaging during the SST activation to inhibit the dendritic activity. So here the red axons are the SST neurons axons. We choose the field where you can see the axons, like in layer 1 or 2, and but there's no soma in the layer 5. So the green is layer 5 neurons. And then right. So first we want to test whether this way works. So we look at the dendritic rule coding first. So for the under control condition, for the significantly rule coding ROIs, when we give photostimulus, the rule coding ROIs coding significantly dropped. Dendritic rule coding could be reduced or with this some activation method. And after that, we want to look at the somatic activity. So the interesting part is for the non-reversing trials, their choice is not rely on rule, but for the reversing trials, their choice is rely on. So when we look at the somatic activity in non-reversing trials, control condition, we put the left and the right twice separately here. So we can see under control condition for non-reversing trials is the higher activity in the left twice and the right twice.
Sorry, your voice is cutting off. It's the quality.
Can you hear my voice?
Yes, now it's better. Yeah, it got fixed.
Oh, it's
I
better.
have a question. Yeah. So did you show us how this SST manipulation affects the behavior?
Not behavior, neural activity.
Right. But do you have
this is neural activity.
Correct. But does it affect the behavior as well, in addition to disrupting the dendritic rule coding?
We try to avoid the behavior. So here we just give the light to one side of the window, and the light is through the two-photon imaging window. So the field is also small. So we want to make sure the activity change is not because of behavior, but just like, and the behavior still not change, we see how the activity changes.
Okay.
So so this is all neural activity. Shown here are all neural activity. So for non-reversing trials under control condition, left trials show higher activity than right trials. But in the photostimulation, there's it's have a general reduced activity, but still left trials show higher left activity than right trials. So the trial selectivity not change for the non-reversing trials, but for reversing trials in control condition, the right choice, the activity is higher than left, but under photostimulation, the choice preference changed from right to left. So means when the rule information is changed, the somatic choice output may also change. So this is an example neuron. And here we have all the neurons. The x-axis is under control condition and the y-axis is under photostimulation. For non-reversing trials, the choice preference not changed, but for reversing trials, like for this example ROIs, and the control trials, they may choose have a preference of choose left, and when it's and the photostimulation is changed into minus means it's changed to right. So the choice when they need the rule, because the animal the neuron need the rule information to decide their choice. So for the reversing trials, this is the trials with same sensory information, but under different rule, and they make different choice. When rule information is disrupt, their choice also disrupt. So these results means the rule information is necessary for the choice output. So the rule information, top-down information reached to the dendrites can influence the coding, the output coding. Am I saying it clear?
Yeah, thank you.
Okay. So we answer this two questions. One is there's compartmentalization, and then the rule information in the dendrites. They are important for the decision output. So now we make a conclusion that the dendritic integration mechanism for rule-based flexible behavior. First, we found there 5 neuron exhibit subcellular compartmentalized representation for rule information in apical dendrites and sensory information in soma, and also globally the choice representation. And the second is the autogenetic inhibition of dendritic activity can degrade the dendritic rule coding and change the somatic choice coding. So with same sensory input, they may lead to different choice output because the rule can alter it even within a single neuron. So that's the take-home message. So dendrites have this computational capacity. So I would like to thank my supervisor, Ning-long, and all my lab members. And thank you.
Thank you. That was great. Did you try the SST inhibition, like the other direction of optomanipulation to see what happens?
Oh, so here I didn't try that, but I tried to image the SST neurons. We didn't do the SST inhibition because we suppose the SST neurons, they target Layer5 dendrites, and usually their function is to suppress the suppress the pyramidal dendrites. And if we inhibit SST, then the dendrites may have more information get into that pyramidal neuron. So like the input information would be very large. So we did not try to inhibit it.
And what did SST imaging show?
Oh, it's really surprising because the SST neuron, they are more engaged than the pyramidal neurons, and they aligned very well to the stimulus onset.
Yeah. And then
Yes.
did you see like rule in terms of rule information, like
Yes, they also show high spleen variance on rule and also sensory. Yes, they're extremely engaged in the task.
and what's the hypothesis about where the rule information is coming from?
Based on our previous study, we think one of the candidates is the OFC, the Yanhe's paper. She's working on the circuit from OFC to auditory cortex. So OFC show strong rule information. That's one of the candidates. We think maybe other prefrontal cortex can also give some rule information.
Awesome. Do we have any more questions? I guess someone just, Javier, you just joined, and I think maybe you got confused with the time. It was 1 hour earlier. I think someone just joined, and like. Anyway.
Yachuang has a question in the chat.
Let's see. Yichun, do you want to ask your question live? So there is a question. Did you guys also do similar 2p optogenetic manipulation for VIP neurons?
For VIP, we don't have that.
Okay.
Yeah.
Nice. That was very, very, very nice. Well, good luck with the resubmission. Do we have any more questions? Sure. That's awesome. Thank you, Yuan.
Nice work. Thank you.
Good night.
Thank you.
Bye.
Have a good day.
You too. Bye.